In part 2 of this chapter, we're going to discuss how prokaryotic cells like bacteria use regulatory molecules and control elements to control their gene regulation. The first thing you need to understand about prokaryotic cells is how they organize their DNA. They're actually really organized. They organize their DNA into things called operons. Now I want you to think of an operon like this. Let's say that you wanted to do a project around the house, and to do this project you need a paintbrush, a hammer, and some glue. Now if you have to look all over the house to find your paintbrush, hammer, and glue before you start the project, it's going to add a lot of time to that project. However, if you know you're going to do this project ahead of time, and you have your paintbrush, your glue, and your hammer all together in one spot, you can just grab your tools and start working. Well, that's kind of what an operon is like. Inside an operon, genes that code for proteins with related functions are put together. So here we have a gene for protein A, B, and C, and they all help with this particular job inside the cell. So what the cell does is it glues together these three genes end to end to end, and then it puts a single promoter and operator in front. So there's one switch controlling the transcription of these three genes. When the cell needs to do that particular job, it can just switch on a single promoter, transcribe these genes really quickly, make these proteins really quickly, and it's boom, it's doing its job. So it's a very efficient model. Let's discuss tryptophan as an example of an operon. And for this, I want you to think about the E. coli cells, the little bacteria that are currently living in your large intestine. Now, tryptophan is an amino acid. Cells have to have tryptophan in order to make certain proteins. Sometimes that tryptophan is available in the diet, so bacteria can just extract it from their food. But if it's not available in the diet, then the bacteria have to make it themselves. In order to make tryptophan, bacteria need three separate enzymes. These enzymes consist of 5 different polypeptide units. So 1, 2, 3, 4, and 5. In order to manufacture these 5 separate polypeptides, the bacteria need to activate 5 separate genes. 1, 2, 3, 4, and 5. The 5 separate genes that the bacteria need to make the 5 separate polypeptides, to make the enzymes, to make tryptophan, are all stitched together within the DNA. In front of those 5 genes, we have the single operator and a promoter. Collectively, we call this piece of DNA the trip operon. Now when the cell needs to make tryptophan, all it has to do is turn on this single set of genes, this single operon, so that it can produce these 5 polypeptides really quickly, and those enzymes can get to work making the tryptophan. Next, let's discuss how bacteria can control this operon. Now, most of the time, tryptophan isn't readily available in the diet. Since there's no tryptophan available, the cells need to make it, and so that gene, that operon, will be turned on most of the time. However, let's consider what might happen if you eat a food rich in tryptophan, such as turkey. If you send that tryptophan down, your little bacteria are going to get very excited. They're going to realize that they no longer need to make tryptophan, and so they are going to need to turn that operon off. Because this set of genes is on most of the time, but occasionally needs to be turned off or repressed, we say that tryptophan is a good example of a repressible operon. Here's what it might look like if tryptophan is not present and the trip operon is turned on. RNA polymerase is going to grab onto the promoter of this operon. It's going to be transcribing these 5 genes. Here's the little mRNA transcript feeding out of the back of RNA polymerase, and it's going to be producing these 5 gene products. Let's say that you've just eaten a big turkey dinner, full of tryptophan, and that tryptophan has worked its way down to your large intestine, and your E. coli have taken it up. Those E. coli are going to realize that they need to turn off the Tryptophan because it's no longer necessary. So how will they accomplish this? Well, inside those E. coli cells are going to be a number of regulatory proteins, and among these will be some proteins called repressors. Now, this particular repressor is produced in an inactive form, so it can't do anything at first. But when tryptophan comes along and binds with it, it activates that repressor. So the repressor can now do its job. The repressor with the tryptophan is going to bind to the operator of the Tryptophan. And look what it's doing. It's blocking that operon. So now RNA polymerase can no longer bind to it and can no longer transcribe the genes. By physically blocking the RNA polymerase, this set of genes has been turned off. In this case, the tryptophan molecule is acting as a co-repressor because it's binding to a repressor and it's helping the repressor repress or turn off the operon. Now, eventually, all of the available tryptophan is going to be used up by the cell. So at that point, it will pull the tryptophan off of the repressor so it can use it. That's going to cause the repressor to lose its shape. It will inactivate and fall off of the operator. Once that happens, RNA polymerase can now grab onto the operator and the promoter and begin transcribing that operon again. So by removing the co-repressor from the repressor, the cells can turn that operon back on. It's the presence or absence of the molecule that the cell needs to make, in this case tryptophan, that actually regulates the pathway itself. So the presence or absence of tryptophan activates and deactivates that repressor, and the repressor can turn the pathway on or off. When there's lots of tryptophan around, the pathway will turn off because it doesn't need to make any more. And when there's not much tryptophan around, the pathway has to turn back on. Tryptophan can also help control this pathway by affecting the activity of specific enzymes in the pathway. So for instance, when there's a lot of tryptophan available, it will interfere with and deactivate this first enzyme in the pathway. If you remember back from Chapter 8, we refer to this type of autoregulation as feedback inhibition. Because the first enzyme in the pathway for making tryptophan is deactivated, it means the rest of the pathway is deactivated as well. And that allows the enzymes that make tryptophan to shut down for a while tryptophan is available. Once the tryptophan is used up, the pathway will reactivate. Because of the presence of feedback inhibition, repressible enzymes and pathways, such as the tryptophan pathway, are usually associated with anabolic processes. In other words, because you're building a product at the end, that product can help turn off the pathway when enough of this end product has been made. Another great example of an operon that works sort of in reverse to the Tryptophan is the Lactose. The Lactose contains the genes that the cell needs to make enzymes to break down lactose. Now, if you remember, lactose is a carbohydrate, and E. coli love to break down lactose. However, lactose isn't essential to the diet in the way that tryptophan is, and it's not always available. So most of the time, there's not going to be any lactose available, so that operon is going to be turned off because it's not needed. However, let's consider what might happen if you suddenly eat a lot of dairy products like milk and cheese, and that manages to reach your large intestine. Well, your E. coli cells are going to get extremely excited, and they are going to want to turn this operon on so they can start breaking down that yummy lactose. Because this set of genes is off most of the time, but occasionally needs to be turned on, we say it's a great example of an inducible operon. In other words, most of the time it's off, and occasionally it needs to be induced or caused to work. This is how the lac operon might look if there's no lactose around and that operon is turned off. There is a repressor for this operon, and in this state it will be bound to the operator. So notice that it's blocking the way of RNA polymerase. RNA polymerase cannot bind, and so this operon is turned off. No transcripts will be made, no proteins will be made from this set of genes. Let's say that you've just enjoyed a big glass of milk and that some of that lactose has made its way down to your large intestine. At this point, your E. coli are going to get very excited and they're going to need to turn on the lac operon so they can break down that lactose for energy. How will they go about this? Well, lactose in this case acts as an inducer. Lactose will bind to the repressor, and when it binds to the repressor, it actually inactivates the repressor. So the binding of lactose to the repressor has the opposite effect of tryptophan binding to its repressor. In this case, it turns the repressor off. When the repressor is turned off, it's going to release that operator, and now that it's not in the way, RNA polymerase can grab onto the promoter and transcribe those genes and make the 3 enzymes necessary for breaking down lactose. The enzymes made by the lac operon are going to break down lactose, so this actually represents a catabolic pathway. Inducible enzymes and inducible operons are often associated with catabolic pathways. So that means that they need some little extra signal to turn on that pathway. And when that signal is removed, the pathway turns off. You don't have the same kind of feedback inhibition that you saw with the anabolic pathways and the repressible pathways. All right, you guys, that's it for now. Make sure you use this information to complete those guided notes and to try all the your turns, and we're going to work with this information more in class. We're also going to learn a little bit more about lactose intolerance, why some people are lactose intolerant, and where the ability to tolerate lactose actually came from. See you in class.